let me start by just acknowledging the Alliance for Regenerative Medicine for serving as a host and providing us this lovely venue for just one more year, I think, before we move somewhere else. So my name is Betty Wu. I will be your moderator for today's expert session on advancing cell therapy manufacturing through collaboration. I also, for the last 3 years, have served as the vice president of our cell, gene, and advanced therapies business across Thermo Fisher Scientific. We focus on fit-for-purpose product solutions in support of the industry. We, and I suspect pretty much everyone in the room, share an aspiration or an ambition to increase the accessibility of these potentially curative therapies to all patients. And among the many enablers that needs to happen to achieve this goal, our thesis is that collaboration is key. And again, I know that I'm not alone in that. So as a technology innovator with a breadth and depth of product and service solutions serving clinical and commercial manufacturing of advanced therapies, Thermo Fisher has actually invested quite a lot in the infrastructure for supporting industry collaborations. Our collaborations can range from technology development to process optimization. We partner in order to shore up raw material supply chain. We also partner at times with clients around sustainable manufacturing practices. So our collaborations are pretty much as diverse as our customer base, and I can say that there is no cookie cutter solution. Pretty much every collaborator comes to us with a unique set of considerations, challenges, and constraints. So we try and accommodate them as best we can. As you can see here, we've brought together a diverse set of perspectives. We have, as you can see from the screen as well, a mid-sized clinical stage biopharma company. Seth Ettenberg, the CEO of Blue Rock Therapeutics, is to my far left. We also have the lens of a large pharma company. So Ivie Aifuwa comes to us from Bristol Squibb. And then finally, in the middle, we have a CDMO perspective. So Dan Herring serves as the general manager for our advanced therapies CDMO business within Thermo Fisher. So nice to have that perspective. Patheon is one of our premier service brands under Thermo Fisher. So as you know, collaborations can take on different shades, as I like to kind of often say, and it can depend on any number of factors from the clinical stage of the company, what their platforms are, certain quality or regulatory considerations that they may need to make at different points in their journey. I say always that it's a two-way street for any collaboration to work, but in some instances, I think it looks more like a four-way intersection. So at times, things can get a little bit unpredictable and hairy, but we can talk about that another time. But I think having a shared vision is a really important first step. I do think that we commonly agree in the criticality of collaborations for together solving the industry problems. So before we kick off this session with a first set of questions that will focus on collaboration, I wanted to start with brief introductions of the panelists, and I'm going to ask them to provide a little bit of context on their respective companies' manufacturing approaches, their platforms, and their pipelines, and maybe as a little bit of a twist and icebreaker that I'll throw in there is just given the current macroeconomy and some of the maybe funding challenges, I'm wondering if you can maybe comment on how your company has maybe adapted your strategic priorities in these kind of times that the year that we're in 2023, as well as kind of moving into 2024. So maybe I'll start with Ivie.
Sure. Hi, everyone. I'm Ivie Aifuwa. I work within Bristol Squibb, and I lead our technology development team within our cell therapy development business unit. So we primarily focus on autologous and are exploring the allogeneic space through healthy donor-derived allo products as well as iPSC products through various collaborations. My team and over the course of this discussion will really we focus primarily on partnering externally and internally to develop various tools that will really support and enhance cell therapy. And so with your question, with the current environment, we are therefore very strategic in terms of the various partners that we want to collaborate with and invest in. So we still pursue various strategic collaborations, we still pursue investments, but we are essentially looking for partners that will enable us to be as disruptive as possible within the space and prioritizing that.
Thanks, Ivie. Dan?
Dan Herring, excuse me, as Betty said, I run the advanced therapies business for Patheon, the CDMO side of Thermo Fisher. So what is advanced therapies for us in PSG? It is mRNA services, so mRNA drug substance, LNP, into the drug product filling. We have that facility in Monza, Italy, as part of our broader facility that does a significant amount of drug product filling. It also is our San Francisco cell therapy site, which we opened in March, which was in the news, and we were really excited about that new facility, which was done as a partnership with UCSF, so we'll get into that from a collaboration perspective, I think, in some of the questions. And then we have a site here in Carlsbad, which does plasmid DNA production at full GMP all the way through from RUO, 3-liter scale up through, we can do 1,000-liter scale in the facility, 3-liter, 30-liter, 300-liter as well. In terms of how we've maybe pivoted a little bit, I think as a service vendor to the industry, we need to adapt with our customers and ensure that we are coming up with solutions to get to what is your real near-term goal, right? So is it an IND-enabling batch? Is it the full clinical trial? Is it a large-scale expansion for your phase 3 going into commercial? And we try to adapt to the price pressures. Obviously, you can say pricing is part of it, but I'd also say that it is about finding that boutique nature at all scales, whether you're smaller, mid-size, large company. I really feel strongly that in cell therapy, you have to treat every customer like they're just starting out, regardless where they are, in terms of that hands-on and high touch, and finding the solution that works for them from a relationship perspective more than anything.
Thanks, Dan. Seth?
Hi, Seth Ettenberg, Blue Rock Therapeutics. Blue Rock is a wholly owned subsidiary of Bayer Pharmaceuticals, run with an arm's length relationship. We are a pluripotent stem cell company, and our lead asset is in Parkinson's disease. But we have a pipeline beyond that in ophthalmology, cardiology, as well as autoimmunity. And so one of the issues that we've run into, and as you asked that question, we talked a little earlier, we are not immune to the outside environment. While we are a wholly owned subsidiary, iPSCs allows you to make any cell type in the body. Unlike what we've seen in the cell therapy companies that have been born before us, where adult harvesting often means one cell type, or at best two, a T and an NK, if you will. We're not making either of those cells, and we're making about 9 different cell lines. Well, you can imagine the effort that goes into as you approach phase 1 and phase 2 for each of those different cell lines. And I think the reality has come home for us in the last 6 months that we just simply can't carry all of that weight on our own. And so we recently had some layoffs, which is a very painful moment for any young company that was growing 50% year over year. And the funding really needs to go towards what we see as a beeline to patients in Parkinson's disease, which is an incredible unmet medical need. And we just recently released our results from just the 1-year data set from that phase 1, and it's incredibly exciting. And it means focus time, and that's okay. We will focus now and bring about 3 or 4 other products, different cell lines, into the clinic. But the funding environment for us has meant many of the nice-to-haves, which were having 2 GMP suites for a cell that you're bringing forward so you could do things in sort of parallel rather than series, have now really come down to often being 1 GMP suite. And we've had to make tough choices like that over the last year, and I suspect that we have a few more years where we're making choices very similarly.
We'll get to the crystal ball in a little bit. Okay, Seth, I'm going to stick with you a minute. As I said at the beginning, collaboration can take on many definitions, many shades, as I like to say. When you think about the 7 years since Blue Rock was founded, can you kind of tell me how collaborations kind of entered into that in a meaningful way?
Yeah, I mean, it's easy, Betty, because it's within our strategic structure and our DNA from the very get-go. The very first years, Blue Rock announced a strategic collaboration with Editas Medicine to be capable to bring forward a GMP and well-validated editing reagent. And so that's part of our strategy and our pipeline. And just about, I would say, every 8 months or so, somebody, I don't know if someone can keep me, check QC me on this, but we announce another strategic collaboration, mostly because we see that in this field, when a product means a device, a product, and an approach, a surgical approach, there are many that have been studying these elements. While we can handle the biology and the cells, the other elements are well suited in other companies that we bring together. And that's been key to our success. We just announced 2 recently this year around BitBio that's using transcription factors to bring forward cell types that we've been struggling on, getting the exact right cell type that we want, as well as Rune Labs and Emerald technologies that are digital health technologies in the Parkinson's area, in the Parkinson's clinic, to collect a much more broad and fulsome data set for the patients and what they're getting from the product. And so it's part of how we think about and how we progress. But I guess I want to take 1 second, which is to say the word collaboration. Many an email will come asking for a collaboration, but in fact, it's really a fee for service. And for me, the definition between the two are collaboration means we don't exactly know how to get there. And we're both going to share the risk and the reward as we get there, and it's going to change as the path goes because we don't know exactly how to get there. Fee for service is, I do this all the time, and I can do it for you very quickly. And I think that key difference of changing the path and being willing to change the path together as you go is really important in collaboration.
I think it's a great point. We've been in collaborations for quite a while, and while I totally agree with you that, kind of it might not be clearly defined in terms of, what the end state should look like, but the path for getting there is not clearly defined. But then when we think about scaling collaborations, we get really sensitive to not having clear milestones because at some point in time, the collaboration may end, right? It should end. And so, you kind of get into this loop of, is it starting to go sideways? And that's why I said at the beginning, to us, having a shared vision is really important because when things start going sideways or, that path starts meandering in different directions, that's fine. But then we have to reset around what is that shared vision and decide if it's relevant anymore.
Yeah. You say it exactly how the endpoint has to be clear, and the endpoint has to be clear, and your ability to traverse, the motivators have to be the same. And you know as well as I, after years of these collaborations, often those things change.
Exactly.
And that's the right moment to think about, okay, have we done what we need to do?
You have to calibrate, right? Great. Ivie, similar question. BMS has been a front runner in CAR T-cell therapies. I'm just wondering where external collaborations have come in to really fuel your clinical programs. So can you comment on that?
Right, so we do a significant amount of development internally. However, just with the capacity of cell therapy within BMS, we've certainly needed to explore collaborations all over the place, right? And so we've done a number of collaborations with Century Therapeutics, GMP Bio to support bringing in novel modalities with various cell types. We have a number of collaborations also with various technology and editing companies to support our pipeline in that way. But to also just backtrack, really with collaborations, it's really strategic partnerships, right? So it's similar to what you were stating. We have a shared vision. I think that piece is super important with, we have a clear direction of where we want to go. We understand mutually that we can't quite get there ourselves, and therefore we're having to support our partnerships to ensure that we're able to meet those goals strategically. Also, with being a biotherapeutic company, we prioritize our clinical output, right? And so being able to have technologies and such that enable us to get there is just critical. And so being able to bring that expertise in-house and inside and learning from our partners has been critical for us.
Well, I think for whatever it's worth, you do it really well. You have prolific partners in the space, and I think it lifts the entire industry up when you have that open spirit to collaboration. Dan, this kind of gets to Seth's comment a little bit around fee-for-service, but oftentimes I think clients come in with a preconceived notion of what the partnership is going to look like, you know, and actually it doesn't turn out that way sometimes, but I'm wondering if you can define, you know, collaborations in your kind of lens as a service provider.
Well, I kind of alluded to it in my introduction, and obviously you are correct. In many cases, fee-for-service is what people are going to end up coming to. But in many ways, I think for cell therapy especially, maybe more so than other ones, that C and CDMO kind of has to be collaboration, right? It's in order to bring any of these things forward, you're going to have to work together. I'd say that the most important and successful programs that I've worked on have been the ones where both parties, the CDMO and the sponsor, have been really in the room together constantly, in the labs too, working together. Now, from our perspective, you can look at it as collaboration, partnership, et cetera. The site in San Francisco is a partnership with UCSF, and I use partner specifically there because we are on their campus. They put in some funding for this site. They have capacity there. We work with them to bring some of their very early preclinical programs forward, and we do in some ways take the risk together in that situation. But within that facility, we actually have something called what we call the Collaboration Center, right? Which is a space that's a non-GMP suite, if you will, that we want to bring our partners in and work with their processes before it needs to go into GMP, work with some of our equipment, the Thermo Fisher equipment that we have available there for people to use. And then the last piece I'd say, and this is unique and special to Thermo Fisher, is that in many ways, your organization, my organization, we are our own partnership and collaboration with access to advancements in the products and the equipment and being able to bring that forward. So in my mind, especially in cell therapy, I think collaboration is the core element of what we do. And I hope that we can continue to make our partners feel that way with every deal that we do.
So I'm going to stay with you, Dan, kind of continuing the theme. We know that stage of the company, size of the company is going to, you know, influence their strategy. And particularly in this environment where bricks and mortar investments become difficult, right? I want to ask you, have you in fact seen a shift to CDMO? You know, being there over the past, you know, I'll say year, maybe a little bit more, just consequent to the economic environment? And if so, how are the collaborations, how have these partnerships changed?
You know, it's not a one-size-fits-all answer, right? I think depending on the company, the runway that the company has, whether or not you have 2 space suites or 1 at this stage, right, drives whether or not they need to come to us. The earlier phase, you have some companies that are just looking to get to IND. They don't want to put any investment internally, and they come and they ask us to get to that stage. You have the bigger ones that are looking for excess capacity, right, for when they get to commercial. So yes, there has been a shift, I would say, in terms of the pricing pressures that exist, which drive us to need to find new ways to partner and collaborate and find an offering that works. But I wouldn't say that across the board there's a drive because of this to CDMO, and I wouldn't say it's the other direction either. I think everyone is looking very hard at their company and what they can do and having to make the decisions for that company on which path to take. And we're very excited to work with all of them, right? I think that you can do the straight tech transfer, and we can talk about that later in some of the other topics, as well as just development opportunities where we can help on the process development side alone and not have to talk about how many batches are you going to do and how long is it going to be and 3-year contracts and stuff like that. It needs to be right now for most people.
Yeah. Maybe I'll ask Seth, you know, just maybe a slight twist on that. So we talked about this in the pre-call a little bit about these kind of, you know, decisions that you have to make and priorities. And I guess, you know, when I think about some of the trade-offs, speed to market versus, you know, the cost of outsourcing something. Any comment there on in terms of some of your strategic priorities that you made, difficult trade-offs? Maybe you couldn't have as many programs. You've been focusing on Parkinson's. So is there anything else that you want to add here in terms of some of these choices?
Yeah, so one, because Dan reminded me, we have at least all 3 models going on in Blue Rock,
Yeah.
and not just one GMP suite, which is why I wanted to correct it. So we have our own GMP suites where we've made choices in Toronto. We used 2 different CDMOs for other products, and even for parts of the process, we're using CDMOs. And then 2 days ago, or yesterday, Bayer announced our fully operational commercial site and a $250 million
Congratulations.
investment
Yeah,
into
Berkeley.
Berkeley, California, into a beautiful location if you haven't been, as well. And so it's not doom and gloom as I want you to point it out, and I still think the science is really supporting incredible optimism, and the validation steps that are coming now outside of oncology are incredibly exciting. And so the investment thesis will follow the data. Today, we're in the choices that you're talking about right now. And so I guess what I'm thinking about is that the time frame, and I liked what you just said around that space that you're creating, because getting into the technologies early on in your process, what machines we're going to use, what systems, having birthed a company out of an incubator prior to Blue Rock, that became essential, both access to those machines because you can't afford them, but then once you start building your data sets on them and learn how to use them, become integral to your process. And so it's a brilliant site that you guys are just getting started like that. And then access to individuals that have been there, done that before. Is really important. We just opened a collaborator space right below Blue Rock for this very same reason, where for our local environment for cell and gene therapies, we're encouraging them to come in for those exact moments, not so much around the equipment, because that's not what we have, but around the know-how and the encouragement to focus their data sets on what they need for the next step of evolution. And I think that part, and it will follow our data validation. We now have multiple companies outside of oncology that are demonstrating the utility of cell and certainly gene therapy at much higher numbers than that.
Ivie, so with the emergence of a lot of new technology coming out for cell and gene therapy, I guess, is there something that really excites you? I hesitate to ask the question because it's been asked like all throughout this conference, so you probably maybe are, I don't know if it's delivery for gene therapies more, or there's plenty of gene editing, excitement for sure, particularly, with some of imminent approvals, hopefully by the end of the year. So just curious, is there anything that really excites you? And the part that makes it fit this section is just, what do you need to see from technology developers in order for BMS, for your team to really decide that, I want to evaluate it, justify, that time and cost associated with that evaluation for adoption into your clinical programs?
Sure. So, with the emergence of cell therapy and with the direction that it's going, extremely exciting. For me specifically, again, going back to my joy and excitement over technologies, I'm really excited about tools that enable us to start considering more adaptive process control strategies, right? And so, within BMS, our core competency still within cell therapy is within the autologous space. And so tools that enable us to start to learn about our process and from there being able to start providing control, becomes extremely critical for us. So it's really important for us to start identifying tools that enable us to learn, yes, but also start to refine our process through integration of various tools within our process technologies and starting to guide the overall product profile over the course of our manufacturing process. So these don't include just process analytical technologies, which are core for supporting adaptive process control, but really any systems, any reagents that enable you to state, based on this particular product profile, I'm going to implement this specific processing strategy. So being able to work with suppliers and vendors to start building that strategy for autologous processing, I think will be important. Now, speaking to, what does it take to support adaptive process control, but also to adopt new technologies as well, it's, I would state that for cell therapy right now, we have tools that work, right? It's not just are we able to support this field, but can we really advance and can we be disruptive in this space? And so when it comes to evaluating new technologies and implementing new tools, it's primarily associated with, do these technologies address key gaps that we're seeing? Are these gaps or these solutions disruptive enough to warrant the investment associated with evaluation and implementation of these tools? It's a significant amount of effort to take a technology, certainly develop it such that it aligns with your processing needs, but also purchase systems to span your manufacturing facility and to support qualification, et cetera, of all those technologies. So it's really, are you addressing a core gap, or are you just moving the needle just a bit? So really ensuring that the systems are sufficiently disruptive.
Yeah, I think in yesterday's one of the workshops, it was on digitization, and obviously that has relevance here, but I'm going to put you on the spot maybe a little bit, but when you think about adaptive manufacturing, right, or adaptive process control, I know that you're applying that to certain parts of the workflow right now, but how do you think about, the entire manufacturing process for autologous, right? Is that like in the 10-year time horizon? Is it in the 20-year time horizon? Yeah. Recognizing that you're already taking steps toward that now, do you ever see a time when the entire process is going to be automated and fully controllable?
Certainly with various tools that are being developed, I do have a lot of confidence that our process will at some point be fully automated, right? Now, in terms of providing adaptive control, it's really how do these tools integrate with the processing technologies at those various
Which
process
includes the
steps.
analytics, too.
Exactly, right? And when it comes to PAT systems, there's a lot of technologies that support more process understanding. We have tools that support items like DO and pH that are more traditional, but there's a fundamental lack of tools that actually interrogate the product, look at cell phenotype, evaluate cell functionality. So being able to develop these tools such that they integrate with the various cell processing formats that are relevant for cell therapy first, then being able to, again, integrate and such that from the beginning of your process all the way through the end of your process, you have clear understanding of the profile and the progression of your product, and from there being able to start building these novel control strategies based on that. So the tools are coming. You do see suppliers here and there starting to put together tools that evaluate and interrogate product profile. So I'm hoping within the next 5 or so years we start to have more robust systems out there to start integrating with our various process technologies.
Great, thank you. So maybe the last question for this little part of the section. Given your distinct backgrounds and roles, can you point to one key takeaway that stands out in your memory as either a really good experience or a really bad experience when evaluating a product or a system, right? Just maybe some learnings for the crowd.
I mean, without naming names, I'd tell you the one really bad experience from technology that I had. This is the decade I spent in Novartis, where we would bring on new technologies, and bringing on a first alpha unit and having it long enough on the bench that we created a whole bunch of data, and the company that started it couldn't support it and couldn't withstand the finances to continue to evolve the technology. And I think that was an important lesson for me about how far you introduce new technology into the greater system, really to create a testing zone, sort of encapsulate that and don't let it find its way through, because eventually projects and or teams become dependent on those technologies, and their data sets and their comparators become dependent on them. And then the financial wherewithal of the backer to be able to adapt the machinery and or even just sustain it from all of the moving parts. And so I think that was a real lesson to me around the excitement around alpha testing, but also the danger to it.
Gotcha. Great. Dan?
So some of you know, I had a background in quality. Most of my career was in quality and operations. And the one that really sticks out to me as I entered into cell therapy was actually around data integrity and some of the equipment that was maybe not fit for that purpose early when it was put in. So an example I'd give was there was a piece of equipment that the password would lock. And if you got locked out, you were locked out, and your process couldn't move forward, and you were stuck. And we saw that in a couple places. There was that piece of equipment. There was some, there was no Part 11 compliance on some CRFs, right? Because the older ones didn't have it. And I think that was the biggest technology growing pains for us, was when you run into that early technology, like you said, that you get something early, and then the whole thing gets built on it, right? So once it's in the process, you're not moving off of it. It's going to be there until you get to whatever change you do. It may be phase 3 later, and you have to work around those things, and it creates so many hurdles. So having a little bit different perspective as you come in, like say with a CDMO, where we've had these different experiences and can kind of say, here's what you're going to face if you use that one piece. But that was probably the one that was most painful to me.
Got it. Evie?
Yeah, and I have a similar experience.
They're all about experiences, by the way. None of you came up with a good one.
Certainly with building our process based on a key technology where smaller suppliers, I personally enjoy working with smaller suppliers for their nimbleness and agility as well, but the financial viability becomes pretty critical. But my actual example is pertaining to the importance really of collaborations with the end users of the technologies as various suppliers and vendors are developing these tools. I really do appreciate that various parties are out there building novel and disruptive systems that will be used to support us, but I can't tell you the number of times where, for example, a supplier will tell us, we're developing this novel end-to-end processing technology, but when you ask key questions, you realize that the technology itself doesn't actually support some of the unit operations that it's anticipated to. So it shows that the supplier, the vendor hasn't done sufficient homework to engage the end users and ensure that the constraints of the unit operations are adequately incorporated within that technology.
Got it.
I mean, I think the positive end of what I'm seeing, at least certainly from the iPSC field, is that as many of us start to make progress, the raw materials, the substrates, the medias, many of the objects that were really research grade stuff is following behind, and the investments are starting to follow behind the progress that we're making. And so the benefit of, as others move forward, there's enough of a market to put the investment in to get to the quality, to get to the testing, and to get to the objects that are needed. And my assumption is for some of us that'll be true in the automation as well. I'm a big fan of not end-to-end. I'm a big fan of unit operations that get daisy-chained together somehow, and as long as we're not in Betamax and heading to Ph.D., I think being able to daisy-chain some of the automation together for me seems like that's where many of us are thinking and heading, and I'm seeing advantages there.
And I'll give one other positive since you called that out. There was a piece of equipment that I've worked with which reduced the processing time, and it was really fast. And so you're doing cell therapy in 12 to 18 hours, right? The whole process end to end. And while that was at times difficult because of the training that went into it, anything that shortens time, especially from an operations perspective, CDMO space, in cell therapy is going to be a net positive experience, right? Because the faster you can get through it, the more throughput you can get, and that's really where we'll talk about COGS later. That's kind of the driver.
Gotcha. So I couldn't have asked for a better setup. This was not planned, I promise you, but based on the poll in the room around, what is the most critical driver for cell therapy growth over the next 5 years, and automation is coming out on top. Last year at this meeting, the theme of the panel discussion was automation. I actually had automation providers on as the panelists, so maybe we can carry this theme forward for the next part of our discussion here. But I think I want to hear from Seth and Ivie. you talked about gaps, that when you're looking at new technology and when you're assessing it, it has to address clear, gaps that you have, that you or the industry has at large. So what are the major gaps that you are seeing right now where automation, you think, could significantly address? So from a quality, speed, COGS standpoint, where do you think automation is really going to move the needle?
Right. So, pertaining to what are key gaps and where we lack as an industry right now, I think that there's a fundamental gap of being able to leverage, going back to adaptive control to a bit, being able to leverage in-process product understanding and being able to actually, again, direct the direction of our processing, right? And so it feels quite often, again, within the autologous space, that it feels like we're shooting in the dark a lot, right? We'll take measurements at the beginning of our process to have a baseline understanding of the cell characteristics, and we'll take measurements, at the end of our process upon release, or with extended characterization, and we treat all the cells the same. All patients, we process them the same, and so it becomes, I think, critical in being able to support items like OOSs and process failures by, again, developing novel tools that interrogate that product profile throughout the process. Again, integrating these tools with our processing technology such that we're able to learn and expand our knowledge about the product as it progresses and as it evolves, and being able to start creating novel methodologies to start to really align to that desired product profile. So to me, that's a pretty significant methodology to start improving product quality in an automated manner, starting to build in our quality within our processing strategy and not necessarily testing for it just at the end.
Got it. Well, that makes perfect sense. Seth?
I think we're not too far off. When I talk to our guys about this, they want inline metabolic analytics of the cells. They want to know what's going on with the cells, metabolism, and how they're performing while they're in the vessel. And then they want simple stuff like cell count without ripping the cells up and hand counting them. They really want inline cell count to know how they're doing it. Those feel feasible. Feels like we're not far. We should be getting there. I wouldn't say that we have exactly what we want just yet. At least we haven't seen it. And then the last is the other end of it when I talk to them is for a non-liquid cell is really around harvest and fill. This is where automation's really got to help. To do the labor on really large batches of cells, we don't want a batch that comes in 1 hour and then 3 hours later because you only have so many people harvesting the cells. And so harvest and fill lends itself really well to automation. And I think that's the other place that we're also looking and playing with that would be incredibly helpful.
Great. So kind of in a similar topic, Dan, there are certain steps in the manufacturing process that it makes all the sense in the world to automate. Other ones, less of an urgency. Maybe going back to your comment around the modular approach. What do you see as one of the most difficult steps to automate? Was the first one that you would say, oh God, if we automated this, it would be nirvana?
I think you just said it, right? Towards the end of the process, that harvest and fill and getting the bottleneck out, right? So you can do all of these different things, automated upstream, the start process, go all the way through, but you're going to bottleneck out if that step takes hours, or if you have to sub-batch because of your CRF capacity, or you're stuck waiting for a manual fill. And a lot of young companies and young processes, it has to be manual, right? That's just the way it is. To get it to where you need to go, it's manual. But to me, that is the spot that I would be really interested in getting to. We've looked at a few heavily automated processes to bring in, and there's an expectation that your COGS are going to go down, your labor is going to go down, you're going to have less attention needed to the process. But then you still have to have the same number of people for that last step. And so you're carrying those people, you're carrying those costs. So if we can get to that place where you're not bottlenecking the end of the process through manual steps, I think that's the most important to me.
I often think about the gown time and the step they're going to do. And if the gown in and gown out is longer than the step they're going to do, we really should put an automation there.
Yes.
Yeah.
Yeah, I'm surprised that like expansion wasn't also kind of high on the list, because lots of high touch, lots of expense at that particular step too, lots of opportunities from adaptive control for feeds and just kind of supplements. So also another area I think that is worth spending some time on.
Yeah, I guess you mentioning that, I mean, it's as basic for me as saying, get me out of a BSc so that I don't have to deal with that complication from a quality perspective, sterility perspective, and I can put more processing equipment into that space and have the processes running in parallel rather than having to stop and put people and time there. So that does fall into that bucket.
Yeah. Okay.
Should help our move into digitization of batch records too. We're there, we're not there, some are there. And I would think as we build in more automation steps, it's so much easier to digitize that than it is a human's experience in the hood.
And Ivie, I know that BMS is early adopters of automation and CMC. I'm just curious, is there a particular?
And you're still only getting a handful of patients out of that batch. Well, is a handful really moving the needle versus one, right? Maybe not, right? Or I've seen some allogeneic, which is still 1-to-1, right? So I think the more patients you can get out of the effort, that's really going to be the thing that moves the needle.
Yeah, and certainly speaking with the various modalities within autologous, being able to ensure the sustainability of this space. Autologous, has been validated within the clinic multiple times to work. It's just extremely expensive, right? And so being able to identify methodologies to really drive down that cost to ensure that it becomes sustainable for patients, and certainly that requires automation. My primary focus is on the process side, but significant amount of automation required on the QC side as well to support appropriate and reduced drug pricing. But also as we're seeing this space really evolve within the allo space and into gene therapy, that there is just the opportunity to go beyond and to attain affordable products, even leveraging similar but more affordable approaches. Yeah.
Great. So I think we have a little bit of time for questions. If there are any in the audience, I think there are the microphone boxes. Any brave soul? Please.
This is one of the most enlightening panels I've attended for myself personally. Sure. Talking about new technology that are coming in and let's say, smaller companies having to vet them, what would a collaboration look like if a company has a new technology that could really improve downstream processing, but they're not large, they don't have the financial wherewithal, let's say, to support everything? What could companies like yourself do when you see this to collaborate with them, and what would you be looking for?
I'm so glad you asked, because I don't want you to mishear me. Mm-hmm. Your breakthrough, your technology is incredibly important to me. The lesson was mine on how much of that, until it's robust and kick the tires and we know it's going to last and your finances are going to last, how much of that comes into the company. So I still think you have to have the space. You have to be a large enough company that you have the space to take that in. And the lesson was on my side for how deep do you let the technology into your own existing workflows and your own existing processes before the moment of I'll say, validity of the technology. But what you're doing is, I think, is essential. I just called out 4, and I think Ivie called out similar things that we are desperate and want someone to make breakthrough in inline analytics, metabolism, cell count, what I think should be fairly straightforward. And it's not going to come from one of us. It's going to come from you. And so I would just tell you that we're going to support that, but it's how we act on it versus what I would say how you're going to act on it. Because you're not going to have the finances until you prove all that. And those are collaborations. Those are us taking on risks together in those moments.
And I'll add, just to answer really specifically, we have something that we're working on where you have to be willing to let whatever you've created really act the way it's going to act, right? So we wanted to do something. It was a good synergy to run some processes, get some good training for our people, and we said, why don't you bring it in? We'll use your product. We'll try it out, right? And there was a lot of, well, can we want to be there? We want to manage this. We want to do it this way. This is what the protocol is going to look like. And the response was, look, we believe in this. We want to do it, but let's just let it go, right? And work in the processes that we have. And so I think that willingness, too, goes back to the shared risk, right, is important.
And just to add to that, from a company like BMS, it's interesting because we take bets, right? We have to take bets on systems that are very novel, and often these very novel systems come from very small companies, right? And so, having the ability to invest in those companies through our investment groups and such has been really advantageous for us as well. But also, we also like to come alongside our partners. We do run into a lot of companies that are coming out with new and interesting technologies, but they also lack that technical expertise to support GMP design and qualification of those systems. So being able to lend our expertise that way as well, such that they can serve, yes, BMS, but also serve the broader industry as well.
Yeah, excellent presentation. I'm looking forward to hear the recording many times. I have a question. Patient material variability. Can you share what you have learned that can help you to adapt your process for patient material variability? What has been the variables that you before didn't consider that now you are seeing that, yeah, we can adjust the process for this? The CD distribution, clumping and clustering, reaction to activation. Can you share some of your learnings to deal with variability?
Yeah, variability. Okay, so what we're also learning is important in driving our product phenotype as well as and therapeutic efficacy is also in core the memory status of cells, right? Whether they're naive state or effective memory state, we see that distribution is important. Also looking at the activation status of our cells has been also quite critical for us. And so now for these items, we test for these at the end of our process. And so throughout our earlier upstream steps, we have no idea really of what memory looks like or post-activation, what the activation profile looks like. So for us trying to start using tools, machine learning model development with various systems to start to understand, okay, what's the profile even upstream? Can we start to refine our activation steps? Can we start to refine our selection strategies? Can we start to look at processing and expanding cells a bit differently based on our understanding of T cell memory as we desire it to be at the drug product state, for example?
Just following up on the comment about locating the manufacturing of the therapy near the clinical site, I just wonder if the panel could comment on innovations needed to kind of bridge that last mile of getting the manufactured therapy delivered to the patient and where innovation's needed there.
We could probably have a whole panel on that one.
You could. I mean, the cold chain logistics of cell therapy are crazy, especially when you get into the fresh products, right? Which there are many still in fresh products. I'll give just a very brief response to it in that I think cell therapy has a need for hyper-specialized logistics, right, at the end. You see a lot of companies, that some of them are here, right, that are doing this. Thermo Fisher has also invested in specialized logistics and cold chain specifically for this. You've seen other CDMOs do the same thing, where it's extending, the services offering is extending all the way out to that last mile, and the couriers themselves. But it's going to be a balance, right? Sometimes you're going to want to be co-located. It's not just the logistics. But I think it's a balance, and I think it depends on the therapy, and I think it depends on the company that is sponsoring that therapy and what their needs are to meet their patient population. Sometimes the right answer is going to be move the patients to where you can get it to them, right? That might sometimes be, exactly. So it's going to be different across the board.
I think when you ask that question, what goes into my mind is what to be careful about. One is, do you want the site to be a manufacturer? And the answer usually is no. You don't want every one of your sites to be a manufacturer. And so you have to, whatever is done there, whether it's hyper-specialized or not, won't be deemed as by regulators as manufacturing versus preparation, or not all products are going to be freeze, thaw and inject. We're lucky with the T cells, but many other products are not going to be thaw and inject for a while. And so with that, you have to be thoughtful of if you have to do hub and spoke versus centralized versus other, until we can get to a place where everything's thaw and inject.
Oh, hi, I'm Karen from Cytiva. I was wondering if you guys could talk about how you view automation and how it can support the manufacturing operators and avoiding batch deviation.
I guess I would just say to you, we're dependent on you for, since Berkeley has announced a relationship with you and a public relationship, and we're really excited about that collaboration. I should have mentioned it in our collaborating models earlier.
I'm glad that you mentioned that, because when we talk about automation, the thing that I take away, and again, the quality background here, is that oftentimes people perceive automation as the solution to COGS. If you automate, you get into these closed systems, you're going to need less labor, it's going to drive down price, right? But realistically, what it's going to do is it's going to increase reliability. I saw, I think, repeated or reliable repeatability. You're going to take those things out of the equation, right? The errors, and that is going to start driving down the COGS, right? The batch failures, the errors, the deviation rates, the things that get to a shorter release time, right? So the more automated, maybe you can drive your release down to 5 days. You can get the product out the door, higher throughput, right? So I think that that's where, in the near term, automation is going to really do the most for us, is by simplifying our operations and thus driving down the peripheral or support costs associated with batches, not necessarily the direct labor to start.
And just to add to that as well, while that is certainly the case, but direct labor through deviation resolution is still significant, right? And so it's not just the process, the robustness of the technology, but also, you know, our ability to enable more of this, you know, automated resolution of failures without having to include operators. So being able to build that within your processing strategy is important. So for various technologies, being able to have a, you know, let's say delta V layer on these technologies and starting to support more automated failure resolution and improving robustness from that standpoint has been critical for us as well.
And clearly, if we study the marketing authorization for advanced therapies, the biggest issue has been batch release, batch failure, and coming back. And so this is where, you said it, but I think this is where across the field we have to do better.
Okay, I think we're out of time. So with that, let me close the session. Thank the panelists for their great insights, and sincerely wish all of you a lot of luck.